Professor Zeng, welcome to join the ACGA virtual seminar series today. Professor Zeng received her MD, PhD degrees and her postdoc training from the University of Pennsylvania School of Medicine. She is currently professor and a director of Shanghai Jiao Tong University Institute of Medical Genetics and the director of the Key Lab of Embryo Molecular Biology National Health Commission, China. She is also a Changjiang scholar and the chief scientist of the National Basic Research Program of China. She also serves as Secretary General of the International Stem Cell Foundations and the Director of Human and Medical Genetic Working Committee of the Chinese Society of Genetics. Professor Zeng's research interests include human genetics and developmental biology, particularly diagnosis and treatment of common genetic diseases, stem cell biology, and the pre-implantation development. She has published in top scientific journals such as Nature, PNAS, Blood Hematologica Development, and has been recognized with many scientific awards, such as the first Young Woman Scientist Award by the Third World Organization of Women in Science, the 10th China Youth Science and Technology Award, and the 5th CC10 Bioscience Innovation Award, and the second prize of the National Prize for National Sciences, China. Today, and welcome Professor Zeng to join us to share her research. Her topic for today is exploration of a novel treatment for beta thalassemia. Welcome, Dr. Zeng, to join us. Thank you.
Thank you for the can you see me now?
Yeah.
Yeah.
Your screen sharing is closed, huh? Well,
yeah, everything looks pretty good.
Now
Why don't you try to advance your slide and see if we can see?
Yeah. Now I see all your beautiful faces. Now I see your beautiful faces. Let me see how I can minimize you so I can see my slide.
So we're gonna mute ourselves for the better quality.
Thank you. Hi, Dr. Huang, I can see you. And
Hi, nice to see you here.
Good.
Well, I think another very important thing I think Hong Ding mentioned is you see that Zeng is an excellent singer. She is really great. And she has.
Okay. Thank you. Well, thank you very much, Dr. Hong Li, for that wonderful introduction. And also, thank you very much for Dr. Huang and Dr. Bao Liming to invite me for this ACGA virtual seminar. I would like to say good evening and good morning and good afternoon to our friends in different continents of the world. And I would like to thank ACGA for all the dedication and wonderful work for the community of Chinese geneticists around the world. And throughout the years, I'm very proud because I think I'm one of the early members because I joined the society since 1990s. So today, since this 30 something years ago, I would like to share with you some of our work for the exploration of novel treatment for beta thalassemia. This is also when I first joined the society, and I think I remember in that meeting, and Lap Chi was sitting next to me, and Anne was sitting the other way, and we talk about the thalassemia work, and this is the more systematic that we did in the past 30 something years since I joined the research. So let's see. Okay, so let's talk about hemoglobinopathies. They're the most common autosomal recessive disorders and occurred worldwide. 7% of the global population carry an abnormal hemoglobin gene, and it is one of the world's major health problems more than 500,000 affected children are born each year. And hemoglobinopathy, it's not listening to me. Hemoglobinopathies include all genetic hemoglobin disorders, and hemoglobin is a protein in red blood cells responsible for carrying oxygen and CO2, and it constitutes of a tetrameric molecule composed of 2 pairs of different globin genes. So these genes, mainly alpha and non-alpha genes, have important temporal and spatial switch, temporal spatial expression in different developmental stage. So for example, during the fetus stage, in alpha and gamma globin together constitute hemoglobin F. And in the adult, the beta globin is expressed instead of the gamma globin gene and generate the majority of the hemoglobin A in it. So in our, it's not good in. Right. So in the mutation or deletion in the alpha or beta gene will lead to hemoglobinopathies, including thalassemia syndrome and structural hemoglobin variants. And when gene defects cause hemoglobin synthesis disorder, this gives rise to thalassemia. So if you have lack of alpha, then you have alpha thalassemia. If you have reduced or lack of beta, you have beta thalassemia. And also when you have structural changes, you have hemoglobin variants like sickle cell anemia, such. And hemoglobin deficiency of the alpha and beta has been the major study for many, many years. And the alpha beta globin, besides clinical treatment, also is a very good molecular models for studying the molecular the mechanism, like the switching and such. So here we have to say one of the biggest in the world, the largest survey, large-scale survey happened in the early 1980s. This survey is organized by the Chinese Society of Genetics, involving about 900,000 people across 28 provinces in China. And then to determine the prevalence of hemoglobinopathies and thalassemia in China. These are some of the Dr. Zeng, Dr. Zeng Huang, and very famous Xu Liang and Qin Wenbing, such. These are the leaders of the group. And so first of all, they met just now. The big picture is in Hainan province, and then the first thing they did is to standardize methods for the molecular diagnosis of thalassemia. Imagine if you have 900,000 people, you have to do the molecular diagnosis. You have to have an SOP or the same way to diagnose, the same way to collect patient. So in the world of large data analysis and in the current world, it is not very say like uncommon, but during that time it's very I think it's pioneering that you have all these people coming from all over the world to come together and also to compare the methods, and then to now they decide which are the one they want to do and how they collect the sample and such. So the Shanghai Institute of Medical Genetics, where Dr. Yu Taozhen and Dr. Xu Zhenhuang founded the first medical genetics. They are just now you saw the pictures of the two people. They're the major leaders of this large survey. So in the first early stage, their survey of hemoglobinopathies in about 600 people in 20 provinces and cities and in all of the China, and with all these different collaborators. And later it became more when the paper was finally published in 1987. They have surveyed about 900,000 people and with just now say 28 provinces. And so far it's still considered the largest survey of prevalence of hemoglobin or any clinical studies in the world to this date. So with that, now we fill the gap of the hemoglobin of national prevalence of hemoglobinopathies in the world. So this this map is filled. And the result of this survey showed a high population frequency in thalassemia. And in during that editorial in Journal of Medical Genetics, it was considered studies on this scale would be extremely difficult in most other countries, and even allowing for the populous nature of China, and the work and the organization involved are remarkable. So with that comment, we have the prevalence of the hemoglobinopathies in China. And we found that the frequency of thalassemia in the southern region of China is much higher than the rest. In particular, the incidence of alpha thalassemia is highest in Guangxi and Guangdong, which in Guangdong also has a very high incidence in beta thalassemia. The chemical structure of 59 hemoglobin variants were identified in about 700 families in China. Among them, there are 20 are new variants, include these alpha globin chain variants, and then these beta chain and gamma chain variants. So these total 20 are the new ones, and they were named after the cities they were discovered. So with all this diagnosis, and we know the prevalence, and today I would like to focus on the beta thalassemia, and also this Zoom thing I cannot get rid of on my screen. Okay, so today I would like to focus on the diagnosis, after diagnosis, the treatment of beta thalassemia. So I was lucky to be involved in the survey in the early parts of 87, but then later when I went to the US in 88 and I joined the group of NIH, and then also collaborating with the Shanghai Institute of Medical Genetics. So we did a lot of study from identifying new variants and to using different ways to do treatment. And to date, we're still developing better ways to do treatment. And then one of the model we use is beta thalassemia. So now thalassemia is a recessive monogenic hemoglobin disorder. And it is a deficiency in synthesis of the beta chain of the hemoglobin. The imbalance of the alpha beta ratio of the hemoglobin heterodimer can produce various symptoms associated with the thalassemia, such as anemia, splenomegaly, extramedullary hematopoiesis, decreased red cytopoiesis, ferric iron deposits, tissue damage, and such. And the highly variable clinical manifestation range from mild hypochromic anemia to moderate hematological disease to severe lifelong transfusion-dependent anemia with multiple organ involvement. Traditionally, severe beta thalassemia patients mostly rely on lifelong blood transfusion combined with iron chelation, and some will later develop a cardiac failure and leading to death. Hematopoietic stem cell or cord blood transplantation are also used as a strategy for partially relieve the symptoms. The resource for finding the matched bone marrow donors are limited. So all of the aforementioned, these therapies are expensive and it can be very painful procedures. So with the development of stem cell induction and gene editing technologies, the gene or gene therapy or cell therapy and autologous stem cell transplantation potentially represent an excellent approach. And we need to focus on the efficiency, safety, and stability and such to improve these methods. So now I showed you that since 87, for the past 30 years, we've been working on with the development of this technology in the world. Then we have about these three areas that we follow the advances in the world. So our earliest attempt were to test the drug therapy or to test the gene regulation or suppression of expression of disease-causing gene or express and such. So the first attempt was to use drug-induced gene regulation in beta thalassemia treatment. So I would, here I would like to tell a story because it all started with My computer's not listening to me. Okay, so everything started in the Thursday afternoon in 1990, April 12th. There is a huge celebration in Building 10 in NIH 10-4 at the Laboratory of Chemical Biology. So the Laboratory of Chemical Biology's former director was a Nobel Prize laureate, and this is the NIDDK, National Institute of Diabetes and Digestive and Kidney Diseases. This celebration was going on because Griffin Rogers, a new fellow, medical fellow, joined NIDDK in Alan Schechter. Now he was the current director of the LCB, and together with Connie Naguchi and such. So they have a very exciting paper coming out from, as we know, like New England Journal of Medicine. Everybody was cheering and exciting. Griffin with Arthur Heinz and all these big guys. So they show that hydroxyurea is effective in sickle cell disease treatment through increasing the production of fetal hemoglobin. So as he was celebrating, and he was saying that, oh, this is great, but this is most likely 3 minutes fame. So he was saying like, this is just 3 minutes fame. Then at that time, during the celebration, was the visiting professor, Dr. Huang, and she was okay. She and Professor Zeng was visiting NIH and the NIDDK, and Dr. Huang was spending a large of her research time doing work with the collaboration of Alan Schechter and Dr. Yitao Zeng. So she has her own lab there. And Alan Schechter and Rogers Griffin was the one new fellow joining. So look at, he was very humble, and he has very ambitious, ambition, and he was very kind at the time and very enthusiastic. So at the time, the two doctors say, no, not necessarily 3 minutes fame. How about we think about treatment of beta thalassemia? Would increases in fetal hemoglobin be useful in treatment of beta thalassemia syndrome by decreasing globin chain imbalance? That's the things they have in their mind. So they look at each other and smiled. And then this is the beginning of a long-term collaboration. So they started, actually I was joining the team too at the time. So we started this in hydroxy treatment in patients. So in Building 10 is this clinical laboratory. I think everybody knows in the clinical, back in the clinical research, they have the drug sent to China, and we picked 2 patients with a severe thalassemia. So one is 40-year-old male who is a lawyer, and number 2 was the 42-year-old female. So these 2 patients has this beta thalassemia called beta 654. I will talk about later. They have severe anemia, hemoglobin levels of 4.1 or 6.45 respectively, and they are transfusion dependent. And they have all these major symptoms like splenomegaly and cytosis and hyperchronia and red cell fragmentation. This is the lawyers. The 40-year-old lawyers, that morphology of his blood smear. So what they did was in the 400 days of treatment, they have a very low dose, say 10 milligram per kilogram per day for 4 times per week. And then later they could have clinical effects, and then they reduced to 6 and such. So it was a very well done and follow-up study with the 2 patients. So now it was amazing. After 300 days, and we look at the hemoglobin and reticulocyte counts are increased after hydroxyurea treatment. So it was exciting that we can use such a drug, which is quite cheap to treat the thalassemia. And even though it's a cytotoxic, so it's cytotoxic, that's why we use a quite low dose to try it. And what we have, surprisingly, is that the hemoglobin F and F cells are reduced post these 300 days of hydroxyurea treatment. show how did the hemoglobin reach, increase. So we found out the increased hemoglobin results from an increase in hemoglobin A. So we increased the beta globin biosynthesis was the reason that you have a higher hemoglobin A, which is a cure, treating the beta thalassemia instead of, when you increase gamma, sometimes they have some side effect. So this is a resultant of a more effective erythropoiesis. And as a result, you see the beta alpha ratio go from 0.3 to 0.5. And with this 0.5, the symptom is much improved. You could see the hemoglobin starting from 4.1 to 6.3, and for the second patient to 9.7. And their absolute reticular counts and reach 25. And the globin chain ratio I mentioned just now. So with this, the lawyer went back to his hematologist and I think in the Changzhen hospital. And the doctor was doing the blood smear and said, no, it can't be. Because this is his morphology analysis of peripheral blood before treatment. This is after. So they both patients have therapy quite well and have been able to resume their improvement. So this lawyer can really ride a bike to a two-hour ride bike to Nanhui district to do to be in court, to argue in court. So he doesn't have to depend on So the second patient also has similar increase in hemoglobin A, which is increase in beta globin instead of a gamma globin, which is a very good sign. So with that, when we publish, and even though this is one of my early papers, I'm very proud to have this as first a medical scientific research.
Can somebody mute? No, thank you.
So today we look at this clinical study, and more than 3,000 patients have participated in clinical studies involved in hydroxyurea on beta thalassemia in different countries and regions. However, they use a little bit different regimen, so that sometimes they, most of them increase in gamma. So we are thinking about recruiting more people, and we're really here because this is an international seminar. We really hope that there are people in other countries and multinational clinical trial could be applied, that we could do the hydroxyurea treatment and to look at more systematically or mechanism so that we could find new targets for treatment. So this is the first studies that I wanted to tell you about. But after we published that paper, Dr. C.C. Tan and others also wrote in the annual review genetics, since 91, Dr. Zeng and his group, his associate at the Shanghai Institute of Medical Genetics, have used hydroxyurea to treat beta thalassemia, and beta globin was increased and was significant in treatment in following this treatment. And therapy at the level of gene expression represent a novel approach. So that was very early using drug to modify the gene expression. So at that time, one side story is, so this 3-minute fame guy, Griffin Rogers. So when I visit him later, this is his portrait on the wall. So 3 minutes fame turned into 30 years of fame. Now he is the current director for NIDDK, National Institute of Diabetes, Digestive and Kidney Diseases. And we start this long-term collaboration. We have many, many papers together. And Dr. Zeng was the first people in China have the R01 and such. So we did many of the hemoglobinopathy studies, starting especially treatment, starting them. So we were thinking that if you can use hydroxyurea to change the expression of beta, can we use other kind of drug to to increase or to change the balance of the what do you say, the alpha, beta, or beta, and especially use of various different. So that's what we did, use of various different methods to change the balance and such. So here we wanted to remind again the pathogenesis of pathology of beta thalassemia and our treatment strategies. So we use this beta 654, I'm going to talk about later. It produces minimal amounts of normal beta globin expression. And then they also, because of the imbalance, there's excess alpha globin expression. So this imbalance of alpha beta globin synthesis really induces very, very severe disease like intramedullary destruction of erythroid precursors and all these very heavy severe symptoms, and ineffective erythropoiesis and oxidative membrane damage and apoptosis of RBC. So what we think was when you have excess of alpha, we could interfere alpha so that they are balanced, or we can increase beta or gamma globin so that they are balanced. So this is what we started by using various methods that I'm going to tell next. But first, I want to tell you about this beta 654. This is one of the most prevalent. It accounts for 18 to 36% of the beta thalassemia, and it has the typical beta thalassemia symptom, except it is more severe. It's a very severe form, and it decreased red blood cell production, anemia, splenomegaly, and extramedullary hematopoiesis, EMH, and iron deposit, and tissue damage. So this was found in 1984 in a Chinese patient in New York by Dr. Kazazian, and which was when I was in UPenn, he was the department chair of my genetics department, where I did my MD PhD there. So this is the most common mutation in the Chinese beta thalassemia. But if it's the, at that time when Dr. Kazazian found he could not detect any beta RNA or protein. So it was considered at the beta 0, which means that the patients would not produce any normal globin. But as we studied, this is one of my very exciting or proud work, and I published with Dr. Shujun Wang and Dr. Yitao Zeng. This is the paper that, it's that changed all the study we have in the later. So what I did all these sequence analysis, and we found out that the 654, there is a point mutation at 654. With that position, there's a C2T substitution. With a C2T substitution and lead to and incorrect splicing, because of the C2T, it generates a new GT dinucleotide, which would, in the context of surrounding sequencing, a sequence would form a parent 5' donor like splicing site, and this in turn activate a cryptid acceptor splicing site at the 579 position. So with that, you will have extra piece of splicing mRNA, which is 73 nucleotides, which is not a multiply of 3. So with that, you will have a very severe and a non-malfunctional of a beta globin. So with that, you have a normal mRNA or a disease mRNA with a longer piece, and which is non-functional. But with our study, with the more, Dr. Huang in the NIH was called the RNA lady and the PCR lady. So with the new technology, we were able to detect about 15% of the normal beta expression. And also with HPLC, we also detect 7% of the normal beta globin. So with that, this beta 0 thalassemia is changed to a subtype of beta plus thalassemia. With that platelet plus thalassemia, and which I was just telling you that we have 15% of a normal beta expression. So we can use that as our model to increase normal, to suppress the incorrect mutant, and to do the alpha beta balance such. And very fortunate or conveniently, Jackson Lab made a human beta 654 model, a mouse model, while one of the murine gene was a murine adult beta globin gene was replaced by a single copy of the human beta IBS2 654 gene. The mouse displays similar symptoms as the beta thalassemia patients and severe anemia with abnormal hemoglobin indices and splenomegaly and related and significant EMH, and as well as iron deposit in multiple organs. And this is blood smears and also messed up. And you can see homozygous beta 654 mouse did not survive, and the offspring of heterozygous beta 654 only have 25% of survival. And the thalassemia symptoms are retained stable from generation to generation in these 654 mouse. So we have this model that we have a very clear understanding of the molecular basis of this human thalassemia. So we could use this mouse model as a exploration of our novel treatment. So from then on, we use this mouse for many, many years, say 2 decades and such. So what we decided is using this mouse model for the normal beta globin decreases that to 15%. We could use this antisense to suppress the diseased 654 globin gene. Such there's a compensation so that it would elevate normal beta globin. And we also use RNAi, which we know that would not totally abolish the expression. So we use RNAi to reduce excess alpha globin so that the alpha globin relative increasement will be reduced. And we use this to restore the balance of alpha and beta globin chains. So now we designed it to use a plasmid vector with a tailwing injection and liver integration. And also with the lentiviral vector, we have a more long-term, say, zygotic or zygote injection. We have a genome integration, and then we can use this to pass on to the next generation. So we achieved an improvement of the overall the alpha beta globin chain balance, leading to a decrease in excessive precipitation of alpha globin chain around the cell membrane, and in turn recovery in phenotype and such. So here we see the for the this is the healthy human. And this is the beta mouse treated with siRNA or antisense RNA. So they both have some effects. And this is the mouse without, so it's, even though it's very, 15% or so in human, it's hard to detect. But with the treatment, we have a correct splicing products. And with antisense and also with the antisense and also RNAi and such, we have a very severe or very significant improvement of symptom. We can see on day 17 after treatment, the amount of phagocytosis plus target cells will reduce to about 40%, and you can very easily see that they're more like the healthy, this healthy mouse or human. And then we have erythroid elements and the presence of myeloid cells and bone marrow is much improved, and the hemostatin was obviously decreased in the red pulp, and EMH is also eliminated in these treated mice, and comparing to these are untreated disease mice. So the number of nucleated erythroid precursors in the red pulp and the pulp mount was decreased and contained fewer immature hematopoietic cells. And the good thing about the mouse model, so you can cut it open and you can see with the treatment, the spleen is much reduced, the spleen size. And so that with this much of decretion, and actually has much better symptom relief. So, and also look at, this is red blood cell apoptosis, and this is with the iron treatment, and almost looking just more like the normal, and and the iron accumulation in the liver and such, and this is, you can see, is much improved. So with this study, we use siRNA and inhibit excess expression of alpha globin and antisense RNA, and then to restore the correct splicing pattern in the beta 654 thalassemia mice. And then we have a significant amelioration of beta 654 thalassemia symptoms in mice by combined antisense RNA and plus siRNA. And then we have alteration of the all these symptoms in bone marrow, spleen, and liver and such. And we have additional evidence for the great potential of these siRNA and antisense RNA as a new drug to develop in technology and gene therapy. So with that, we have published and such, and receive a well claim and say this is a different approach to the treatment of beta thalassemia. And this is an interesting, attractive experiment system, and easier to evaluate absolute erythroid specificity of the global cassette, and the integration of only a few vectors can into a single embryo cell reduce the risk of insertion mutagenesis. So with this is also very early work, and we also talk about now because beta thalassemia is because the imbalance of alpha and beta. But only after our work in RNAi and antisense RNA, this is the well-established model that people now talk about how to continuing or better improve the alpha-beta ratio, so that there's a better balance, so that we could improve the symptom, alleviate the symptom. So I talked to you about the gene regulation with the medicine, with the drugs, or with the induction of these normal genes and suppression of abnormal genes. But with the technology advancements, we were thinking about how do we modify the gene itself, so that if it produces abnormal genes, how can they be corrected to produce normal cells? So we turn to the gene editing, and it could replace disease-causing genes and repair disease-causing genes, or delete disease-causing genes and such. So this is the more recent work that gene editing for therapy of genetic diseases. So we have genome editing technology. We're very, very aware of these. This has been, very recent, and it's also, everybody knows about the ZFN, the talons, and CRISPR-Cas9. So as we move more and more towards CRISPR-Cas9, we found out one thing, that with talons and CRISPR-Cas9, for the CRISPR-Cas9, it has to have a PAM sequence, or the guide SG guide RNA. But with our 654, that C2T mutation, we could not really find the relevant or the ideal PAM sequence. So that we turn to talons, which is not as efficient, but then also it could be treatment efficient. So we designed the talons for the beta locus, and also to generate talons transgenic mice by microinjection. And we produce the, we screen the talons mRNA by RT-PCR, and we screen the transgenic founders with southern blot. So from the RNA and the protein level, we have the talon, the founders. And then we look at the treatment. Before we look at the treatment, we look at the molecular, the RNA splicing, which is one of the most severe or significance of this correction. So here we have the talon beta, and this is the beta 654, this talon beta treatment. as you can see, this extra band, another, again, this correct splicing in compared to the human data, which is shows the RNA level, there is the correct splicing. And then we also see it in the protein level. So now we have a very good proof that we corrected this order and molecularly. And from the hematological analysis of mice, and we also see the RBC and hemoglobin and the reticulocytes and such is also improved. And then we have the smear and such in the poikilocytes and reticulocytes and reducing these treatment mouse. And once again, we look at different, histologic proof evidence. And so this is, I would say, the sacrifice these mouse for the human mankind, the greatness of human mankind. So you can see this is even more severe, significant than the earlier study. So the talents of beta 654 has a much smaller spin, and this is almost normal. So the spin size and coefficiency in the mice much improved. And most recently, we suddenly realized that instead of the 654 loci, the C2T, and since it activates the cryptic splicing subsite into 579 I told you about, why don't we try to see if we have this availability for CRISPR-Cas9 modification or gene addition and with this loci? So yes, so we have once again find a way to do CRISPR-Cas9 to treat beta thalassemia using the mouse model. This is the treatment strategy, which is almost similar as all the before in analysis of genome editing, the efficiency of the genome editing, and then for the treatment or the improvement of the beta thalassemia syndrome and RNA, PCR, RNA level and the protein level, and also the other symptomatic and such. So what we see is we have the correct splicing ones, and we have the protein Western level and HPLC and such. It's not moving. I don't know why. Is it moving on your side? Your end? No?
Not moving.
Let's see.
The other way is you can
It off and
close,
then-
close it and then reopen. Yeah. Or you can stop the slideshow.
again and then start again. Okay.
Let's see.
Stop sharing and start again. Hi, Dr. Ball. I saw you now. Did it go?
Are you Sharon? We didn't see your screen.
Let me see if it's the right. Oh, okay. Can you see now?
Yeah.
Yeah.
Oh, thank you. Okay. So now with the new editing technology, so we have CRISPR-Cas9, we say the hematological analysis indices for RBC and hemoglobin and the reticulocytes. You could see from this is the disease mice. They have red cell, 6.4 times 10 to the 6. But then this is see that our treatment to CRISPR-Cas9 is almost back to normal. This is very normal, higher than normal. So we have the again the blood, bone marrow, spleen, and liver and such for the pathology and the histopathology. So these are, when you look at it, much like the normal. And the spleen size is also much improved. The splenomegaly is reduced. So with these gene editing for treatment of a beta-654 thalassemia, we have telemediated deletion, the mutant site, and the CRISPR-Cas9 strategy restore the correct splicing pathway to generate normal beta globin proteins in gene-edited mice. The clinical symptoms, including hematological parameters and tissues pathology of all of the edited beta-654 founders and their offsprings, were very much significantly improved. So this is a one that you could pass on with generations. So this is not just this generation or improvement or treatment. So the survival rate with the gene edited heterozygous and homozygous beta-654 mice were much improved. In a few minutes, I'm going to tell you this. All these studies have a much better survival of homozygous. Our study demonstrate a new effective gene editing, especially with the CRISPR-Cas9. We provide a new idea or new approach to not only to correct disease mutation, but you could to correct the related mutation. So finally, the next few minutes, I'm going to come back to the gene and cellular therapy, because what I told you in the gene regulation or induced expression modification and the gene editing on the inside to correct the disease. And now we could use stem cell or gene to modify the stem cell that we could do what I told you about the lentivirus to modify the stem cell, or we have the most recent IPS, and as I was one of the early people in China to work on the IPS, and we have a gene modification, and this is also very well received when we use IPS to correct the 654 mice, was also published in the Hematologica and such. But today, the last few minutes, because of time, I only want to tell you about the in utero transplantation injection. Is I just told you about this gene editing that it could be passed on to next generation. And this one is the model for prenatal treatment. And it also has easy homing and continuing treatment after birth. This is the for us, in China now with the say the opening of or realization of the for the birth of the second or third child. And lots of people were high, age or the more severe. If you have genetic diseases, you cannot just have the luxury to have another or have abortion and such. So in this term, it will be the future that you repair these disorders, or you have this in utero prenatal treatment of some of the genetic diseases. So this is last I'm going to tell you about the exploration study on prenatal treatment of beta thalassemia by in utero transplantation, stem cell transplantation, in utero hematopoietic stem cell transplantation. And it is a new stem cell transplantationology, as the immune system of the fetal goat is not mature at the early stage. It will not produce immune rejection reaction to transplanted exogenous cells, such as human hematopoietic stem cell. This work we first did in the goat. This is Dr. Xuzhen Huang and me, and this is Dr. Yitao Zeng and Huang. So this model was considered very the pioneer model and by Science, one of the editorial. And so we have the stem cell in utero transplantation. We could do normal chimera goats and study. We could do of the cancer or stem cell or blood disease models. We also could do liver injury and also beta thalassemia model. So these are disease model that we could use normal stem cell to treat. So we first studied this in the goat because it's a large animal. We have more cells. We have transgenic human hematopoietic stem cells. We injected in either laparotomy or Dr. Huang can perform it non-surgically under guidance of ultrasound. And then we have to deliver the intraperitoneal, the stem cells into these goats in utero during the first equivalent to the human first trimester. And we look at engraftment, survival, plasticity, and multilinear generation, and also with again this molecular analysis. We found there is an engraftment survival expansion in these goats that human stem cell
genetics.
I'm sorry. Human stem cell can survive in the chimera, and also we have a second generation, third generation, and we have also these different tissues of hematopoietic tissues that will express this human cells. And then we have these, say, goat model. With this experience, we could use it as the beta 654 model. So as you know, like the mouse is very small, and we developed new tools, new technology to do the stem cell camera mouse. And we have a stem cell hematological parameter analysis, observation of stability of the treatment long-term. So this is the mouse that we exposed, and these are all the little fetus that we injected at 13 days, and we injected the human GFP plus labeled so that we can trace them. So this is the mouse that after surgery, and we look at camera analysis and omics and functional analysis and such. And we can see in the SCT stem cell transplanted goat, you can see in the peripheral blood the transplant, these human green cells. And then you have these routine analysis of peripheral blood, RBC, and the hemoglobin and such, all much increased. And then the reticulocytes and such, all very improved. And the red blood cell indices is also increased, and hemoglobin level in the peripheral blood also increased. Okay, can you see now? So with this treatment, and we could very obviously see again, we look at the histology, EMH in the spleen and liver was much improved, and we are very excited that if we could do this, it's not going again, huh? Why? Okay, I'm almost at the end.
Go back. Okay.
So now this is spleen size, in which I show you. This is also these the routine thing. When we have the patients with all these severe diseases, severe symptoms, we look at these symptoms, see how the mouth is enough. Okay, so with the chimera mice, we generate the prenatal model to treat these beta thalassemia mouse. And then we have a stable long-term chimerism and correct EMH, and also reduce liver iron accumulation and maintain RBC indices up to what we look at 250 days. We use this to humanize the mouse thalassemia model that to investigate stability of therapeutic effects. We're currently also looking for partners to work with these OBGYN departments or hospitals. Hello. Hi. Okay. So in summary, we have use this beta 654 thalassemia mice mouse model that we have all these symptoms of the human severe thalassemia symptoms, and we use this treatment with the imbalance treatment of the imbalance of alpha globin and synthesis, alpha beta globin synthesis with RNAi antisense, and we have lentiviral vectors and to deliver the genes, and we have talents, CRISPR to gene editing. We have IPS for the a correction in the stem cell level. And then also we have the stem cell in utero transplant prenatal. And then we have a treatment effect that is very obvious significance because we have live birth homozygous with double integration of the beta thalassemia mice. So each methods we all yield homozygous mice, which was not born in naturally. So this is with the different methods, it has a different kind of efficiency. But the availability of the homozygous and the increase of the heterozygous from 26% to about 50 to actually 70 for some of these studies. And it was a very much improvement. And these symptoms remain stable and from generation to generation. So in the past, the 50 minutes, and I was telling you about how the survey that I participated in as the, I think I was junior high student at the time, and we participated in this greatness of the survey. And then later used one model. I worked in the NIH with Alan Schechter, Griffin Rogers, and Dr. Yitao Zeng's group and Dr. Xu Zhenhuang, and to discover the new mutation and use that and then use different ways to treat the thalassemia. This is applicable for different scenarios. And later we were generally focused on genetic diagnosis to improve diagnosis and gene therapy and the mechanisms of genetic diseases. Use beta thalassemia as one of the most obvious model, but also we work with other genetic diseases, in which I don't have time to go through. And we have diagnosis and therapy and mechanism that we have a future that we look for collaborations. We look for like in the past 40 years in the international collaboration, academic corporations, very important. We have many international conferences, which is the leading conference or the first in China, in the world, that bridging the West and the East, which is very important, I think, as ACGA, that we would also want to do that. And now in the past, we have worked with 60 universities around the world in all these different continents. And for me, my story is in the 79 when the Dr. Yitao Zeng and Dr. Huang was establishing this first Institute of Medical Genetics. And I was very lucky to be involved or be involved and not actively involved or and this is the papers, and we published in first papers. And this is a lifetime dream to become doctor. I want to, in the last 2 minutes, I want to share with you. As hemoglobin brought my parents, Yitao Zeng and Xu Zhenhuang together, which was described of the first hemoglobin, abnormal hemoglobin M, which is a symbol of the marriage of Dr. Yitao Zeng and Dr. Huang. And that's when I have a long lifetime dream to become a doctor. And I was very fortunate that when I was still a youngster, dreaming to become doctor to cure disease and continue the great humankind, to cure human disease. And I have appreciated the opportunity I have more and more each day that to be touched by some of the greatest souls in the history of human genetics, one of these being Dr. C.C. Tan, and throughout my career, and he has been an encouragement. I know he's also encouragement, a father figure to my father. And for this, I also met many of the great geneticists and in the ACGA and all the previous presidents and such. And as a junior high student, when they found the early training, I had very lucky to be early training at the first medical institute in China, medical genetic institute in China, Shanghai Institute of Medical Genetics. And as I was growing up, I was learning lab techniques and being dreaming to have a career in medical genetics. And I have very fortunate to have a father figure in a scientific career, Professor Yitao Zeng, who has leading me and it showed me the responsibility, the passion, the patriotism, and such So I was very, very excited and fortunate, even more lucky to present or to introduce Dr. Yi Zeng just a half year ago, and he received this Lifetime Achievement Award by our society, the Association of Chinese Geneticists in America. And with that, he's not only a father figure for me in the scientific world, but also in real life. So today is in China is the Father's Day. So with that, I really want to wish Dr. Yi Zeng happy Father's Day, and also to those that are the great teachers and father figure to us researchers, and to show us the passion, the dedication, the patriotism, and how to become a great scientist, and even more so an extraordinary, great human being. And this is, I think my father would appreciate these doctors that trained him and showed him, and he continued showing me, and I'm going to continue showing to our future scientists in our institute. And with that, we look forward to seeing you, meeting you in Shanghai, Shanghai Institute of Medical Genetics. So here's all the funding and such. And I really want to thank you for giving me this opportunity to share my life story with you as one of the luckiest geneticists in the world. And thank you.
Thank you, Professor Zeng. This is a wonderful presentation, not just the scientific and knowledge, but also your life journey, and it's very impressive, and will be inspire a lot of young generation continue follow. Yeah, I do have a question. So it's interesting, and when we were in training, and everybody started routinely use hydroxyurea to treat a sickle cell, and so the mechanism, as you said, is increase the hemoglobin F. But what's the difference in beta thalassemia? And I am so impressed in the observation was not to increase the hemoglobin F instead of increase the beta production. Is there any explanation and between the difference?
This is a very exciting area that I want to go into, and also I want to include more people from all over the world, because currently there are still many people who are in the clinical studies for hydroxyurea. So I was looking at all these clinical studies. What we did was, at that time, because of Dr. Huang, Zhuzhen Huang was working at NIH, you know, as I said, Dr. Yi Zeng and our SIMG has the long-term collaboration with Alan Schachter and lab and the LCB of NIDDK. So they were celebrating, and they were, you know, just exciting. And then they just say like, oh, you know, this sickle cell thing, and how long would it go? How far would it go? And these Dr. Huang and Dr. Zeng, they were looking at each other, and they say, why don't we try it? Because for beta thalassemia, if you increase gamma, everybody knows, if you increase gamma, then you can treat, right? So the alpha beta balance. So with that, we went back to, you know, they went back to China, or Dr. Yi Zeng went back to China, and they were doing this clinical research. Because we're very, what would you say, worried because it's a cytotoxic drug. Mm-hmm. It's a cancer drug.
Yeah.
So what would they do to someone already have anemia? And especially if, you know, gamma would be good for beta thalassemia and such, because it's the, you know, the thalassemia, not the structural events. So they started with very, very low dose. So they used very carefully select these patients, and very surprisingly that we only increase beta instead of the gamma. So we also think it's because of the low dose. So now this is very exciting that I want to see if anyone else in the world, and when they do, you know, these 3,000 patients, would they have, you know, this beta and gamma? So far, we haven't heard from anyone or, you know, read any reports. So this is one of the major projects I want to do. So now we have the IPS, we have more of the cellular models. We can have a patient's IPS or cellular, we can treat it hydroxyurea. We can see how the low dose and high dose, how it affects, would that turn on a switch on some other genes instead of BCL11 that, to increase the, to work on the gamma. So can we actually use beta, therapy to treat beta thalassemia? So this is even better. So your question is also my question, but I think we really want to recruit more people who can help us, and then we would do the more molecular. As you know, like our study is from molecular to clinical and then to applied science. This is the future that we wanted to treat patients. We wanted to be helping patients. So with that, I think we would design.
Can you mute?
You can mute them.
I don't
Yeah,
have.
Anyways, I'm almost
I'm
done.
not,
So
okay.
here I'm going to say again that can someone, if they're still doing beta thalassemia research, and we would like to work together, we would like to collaborate.
Yeah, it was just amazing.
Thank you for the question, and this is something we are also excited about, because during that time we could really find more targets, maybe more regulatory targets. So now after 35 years, we came back to the hydroxyurea because we have more models, more technology that we could use. So it's just like in Chinese we say 螺旋式上升. So this is something that that we were very excited about. And I'm looking at, I'm growing old and old every day, and something, like my father, Father's Day. And really, thank you, Dr. Tolimi, for allowing me to present this on Father's Day, because this is really great figure, great gift I can give him because we were locked out for so many times. So thank you.
Is he here?
He's online. He's online.
Oh,
So,
yeah. Happy Father's Day, Dr. Zeng.
So, okay, I have a question. You see, well,
yes,
that's a great talk, and obviously you have a multi-copy of a science gene in your DNA. Okay, so regarding you see follow-up, you see Hong's question regarding hydroxyurea, and I assume this is another mutation-specific induction for beta chain, right? You see, because you see a mutation, the model you talk about, the patient you use is mutation at you see 654. That mutation is almost a non-mutation, right? You see pretty much no expression or very minimal amount of beta glutamine. And do you see a similar phenotype? You see if you use a different mutation like a missense mutation, like instead of a similar nonsense mutation?
Yeah, I would like to look at that because we can produce some beta, even though very little, even 15 or 7%, but that's kind of cheating, right? So that's kind of because the beta is normal. But then would that do with the other ones? That would be amazing if you could combine hydroxyurea with the gene, maybe editing, editing technology to make it safer, and then later, I don't know. We have all these tools, we have all these combinations, we can play, and I think the future is great.
Okay, another question is the efficiency of you see your gene editing. You see a Talon or CRISPR, so. In your case, you are not using a stem cell. You see, like a CD43 positive cell. You correct.
I'm also using stem cell because I don't have time. So now today I skipped the stem cell part, even though I have a major affiliations and with a stem cell. But today I wanted to just talk about molecular. And then in utero basic touch. So we do with the stem cell. But the results was kind of amazing because for the CRISPR-Cas9, it has a better improvement of symptoms.
I think you see CRISPR, when you use a CRISPR technology, did you try to destroy the internal splicing site? Because you see when you CRISPR, you pretty much non-homologous recombination, and then so you probably will try to remove either you see mutation site or you
Right.
try to remove, alternative splicing site. You can do either way, will work, right? You see, because you by destroy 579, you pretty much remove alternative splicing. If you can correct the mutation, you see 654, you pretty much restore the wild type. And so you can do, I assume you can do either way, you see a similar effect here.
Yeah, but we want to increase better, the accuracy or off-target effects. So right now we don't see much of the indel and such. So we have a very detailed analysis, but still that with these technology, we still want to improve the efficiency and the safety and as such. So there's still a way ahead.
Yeah, that's great talk. Thank you.
Are you plan to move to the clinical trial? I saw the alpha thalassemia was kind of active in the clinical trial website. They're not actively recruiting, but it is listed under the clinical trial website for the intrauterine treatment for alpha thalassemia.
Yeah, I think alpha is quite hard. Lots of people talk about, should we do alpha thalassemia? And this is quite hard to do, alpha thalassemia. So for the beta with the different type of mutation, and it's easier that you have all these different beta non-alpha. So this is, I think it's easier with the beta. So right now with my plate full, I'm going to do beta, and then I want to recruit more people to do beta with me. Yeah.
Wonderful.
Your question was very interesting because during the time when we published that paper, and it was just everybody was raising the question, why do you, increase beta? and everyone else was doing gamma, but we just do, this is our results. We cannot really, change it. But in the future, when we know the mechanism, and then we would have a better understanding, and maybe the more than, Dr. Huang was mentioned that the more future and more opportunity and possibility.
That's
Thank
wonderful.
you.
So, Fanyi, I just want to quick comments. I know for the sake of time, I just want to thank you for your amazing talk and share your wonderful journey, not only you and your father and the mother and your whole family. So great contribution to the field of human genetics and to in China, not only in China, also to the world. And so I know Dr. Zeng Yitao, or Professor Zeng Yitao has been a great father figure for you. And he has also been a giant and mentor and, the great scientist to many of us. I just want to thank you. And using that specific occasion, happy Father's Day to Professor Zeng and your family. Thank you so much.
Thank you. Thank you very much.
Yeah, thank you. So with that, and we may just end the seminar. Next week, actually, we have Dr. Yu give another great talk at Joint Reproductive Society with ACGC. So the same time next Saturday night, and yeah, meeting you there. Thank you.